habitat, but they used anything that I gave them, even pieces of dish like sponges that were like brightly colored, they used. And then always, they placed materials around their movable parts. And so I don't think it looks like a crab. When I see this guy who's free of decorations, he looks like a crab to me. So if that's my search image, if I'm a predator, this is easy. This one doesn't look so much. Is everybody good? So that's kind of evidence for disruptive coloration. So now I'm going on to grooming, which is kind of like my baby. I love grooming. Okay, so with grooming, I have studied so much on shrimp behavior grooming. And then I realized that no one has looked at how crabs groom, right? So that's kind of a big deal. And so what I'm doing is I looked at how crabs groom, and specifically using spider crabs as a model. And so the reasons I use spider crabs as a model is that spider crabs have these special decorating CD that blue crabs don't have. For example, stone crabs don't have it. And so I'm interested in, do they groom the regions where those decorating CD occur? I'm interested in what appendages they use, because crabs have totally different appendages than shrimp. They have totally different body regions. Their gills are enclosed in this basically bricked-in wall, and that they can't actually groom their gills, whereas shrimp can groom their gills. And so I was interested in what is the time budget for grooming for crabs. And if I put these crabs in different environmental situations, which means I give them the capability of mating, I give them the capability of fighting, how does grooming change given those pressures? And then I want to compare it to what has been found in lots of different shrimp. And so my hypothesis is that I'm predicting that crabs will groom the regions where they have decorating CD. It makes sense, man. Let's protect these things. If these are the things that are protecting me from getting eaten, I want to keep them free. I'm also predicting that large crabs will groom their decorating CD more. Why? We know that they're going to undergo a terminal molt. You think they want to protect those decorating CD as long as they can. And then 3, I think that their budget is going to decrease given the capability of interacting with another animal. So my methods, just to tell you, I did 4 different studies. I did one study which was a 24-hour study, which means I sat in a lab and watched crabs for like several 24-hour periods. And what was determined was that the crabs groom the same in the day and the nighttime, first of all. And that data I'm not presenting to you, but crabs groom pretty much consistently, whether it's nighttime or daytime. And then I also did another study that was called Social. In Social, there were 2 crabs in an aquarium. There was a divider. They could see each other, they could smell each other, but they couldn't touch each other, okay? And I'll present some of that research. And then I did behavioral and agonistic, which I'm going to go over. The behavioral was I wanted to determine the time budget for the individual when the individual was by itself, thus its time budget should be the highest. I wanted to determine what body appendages were groomed, what appendages actually were used to groom. And what I did is I looked at 142 crabs, okay? And I observed them each for 30 minutes. And I recorded whatever grooms they did, how long they groomed, how frequently they groomed, and I had to use a voice recorder because sometimes they would groom so fast that I can't write that fast, so I would use a voice recorder and transcribe it later. The crabs were not fed within 24 hours of the experiment, and if they molted, they weren't used within 7 days of molting. I recorded if they picked, if they brushed, if they scraped, and what appendages used it. This is a mock setup. So one spider crab was placed in an aquarium. The water was constantly being filtered. They didn't have a habitat to hide in. There was no food present. And they also did not have any access to other crabs, and they were visually separated from other crabs as well. So the agonistic was the fun one. The agonistic was where I put 2 crabs together and I watched to see what happened, okay? So, and we had lots of different behaviors that happened. So what I did was these crabs were size matched to eliminate variables of size. They were within 10% difference in body size. And what I did is I looked at 45 different pairs. I looked at 15 male-male pairs, 15 female-female pairs, 15 male-female pairs. And these crabs had never met each other before. They were novel to each other. And what I did is that, again, not fed within 24 hours, all that good stuff. And clearly I observed these crabs for a long time too. So in this experiment, this is showing you the 2 crabs. The same things were recorded. But with this one, how much did they fight? How much did they display? Did they mate? Other behaviors were also recorded as well. So now if I go into the results, the first thing is this is what the spider crab looks like. These are its eyes, the rostrum. These are the first chelipeds, which pinch themselves. And these guys are the mouth parts. How crabs work is that crabs have paired mouth parts, okay, and they have 3 pairs of them. They have the third, second, and then the first, and they're paired. And what's really weird about crabs, which most people don't know, is that when crabs move their mouth parts, they have an extension of the appendage that actually goes inside their body and moves over their gills and cleans their gills. So when crabs go like this, they're actually cleaning their gills, okay? Does that make sense? And so this is a video that shows you this crab, and this was not taken during the experiment because there's clearly food floating around, but you can see when the crab is going like this is his third one. When he's going like that, he's actually cleaning his gills. These third, there he goes, he just cleaned his antennae. So they have this little palp that kind of reaches up, he reaches down and cleans his antennae as well. So all those motions, guys, are actually the crab grooming. And the other thing that's really cool about crabs is that how does water get into the gill chamber? Is this opening right here that you can see? This is where it goes in and cleans its gills. The water goes in this area, and we'll talk about that a little bit more with morphology. So I wanted to know, do large and small individuals groom themselves the same? Remember, I predicted that large individuals would groom their bodies more. As you can see from this nightmare mess of a graph, there is no relationship, okay? It doesn't look like anything. And with an absolutely horrible R-squared value, there doesn't seem to be any relationship between large and small individuals and their grooming frequency. And this was grooming frequency. So people never get this. So let's see if we can get this. So I can do a groom real quick and groom my antennae. Are y'all good? That's a fast groom. That's once. However, and that is what we call like a 1-second groom. However, I can go and I can really groom this region and clean it. And that's only considered 1. But the time that I invested is different. So when you look at grooming, you have to do frequency and you also have to do time. So this one was frequency. The next one is time. Again, horrible R-squared value. There is no relationship between small and large individuals and the number of times or frequency that they groom each other. So the deal is that it's totally not what I predicted, that large and small individuals, roughly they kind of groom equally, that there wasn't the relationship that I thought was going on. So now if we look at what grooming appendage do they use the most, they use that 3M to groom. It's really unbelievably, their mouth part is the use grooming appendage. They use it by going like this. They clean their antennae. They can clean their other mouth parts as well. And so what we can see here with the p-value less than 0.05 and A and B just represent that there was a difference statistically, that they do use their third maxilliped more. And what's really cool in crabs, guys, is that I don't know if y'all know this, but crabs are usually handed. So you know how like we're left-handed or right-handed? Shrimp and decapods are considered usually to be handed, that sometimes that they use one appendage more than others. And so what I did before I combined the cheliped, the first pinchers, is I looked at, did crabs use their left one more than their right one or things like that? What I found was that they used them equally. So there doesn't appear to be any handedness in this crab, which is kind of odd. So now we're at body regions groomed, okay? So I'm going to work you through this graph. First of all, this is frequency. These are body regions. This is the most anterior to the front of the crab. That's the back. So guys, this is the rostrum where they decorate. The left and right pincher. The third, second, and first maxilliped. The antennae, which are sensory. The eyes, the gills. There's this ridge along the crab that they decorate. This is the mid-carapace region, the abdomen, and then other walking legs. And so remember, what I predicted is that crabs would groom regions where they have decorating setae. The yellowed regions are the places that have decorating setae. We can see they're not decorating them very often, right? And we can also see that what is predicted in other decapods, that they groom regions for sensory and respiration. And those are what the green asterisks are, and we can see they do that. So they seem to be grooming regions that are sensory and respiratory related more than decorating. So now I'm going to go to time, the same axis, okay, but this is time in seconds. Again, we predict this. Clearly it didn't happen. Instead, we have them grooming sensory and respiratory structures. So what I really wanted to do is I wanted to do a statistical test on this. So what I did was that I combined all the sensory structures and then all the decorating structures. And I wanted to do a comparison, and I found with a very significant p-value that clearly there was a difference, that they're decorating their sensory and respiratory structures more than their decorating structures, which is, again, not what I predicted. And this is consistent, though, what's seen in other decapods. Respiratory and sensory structures are way important. So now we get to the fighting part, okay? So this is time, and this is what they did. So we have the reason that feeding is low, remember, is that they weren't given food. So why is there anything with feeding? Because shrimp, crabs, they're always going around picking up things in their environment, okay? And so what we saw is that statistically there were differences, and the different letters represent that there were significant difference. So, for example, crabs, for the most part, they fought, okay? The next thing they did is it tried to mate, okay, which is really weird. Even like two male crabs is kind of odd. And then, actually, it's not odd. Decapods do that all the time. And then we have grooming, and grooming is really low. So we put these crabs in an environment where there's another individual, and their cleaning basically goes way down, guys. And so grooming clearly decreased when other primary actions were present. And so grooming doesn't appear to be important when other individuals are present. And this has really huge impacts, guys. And so then what I wanted to do is I wanted to look at the time budget. And I wanted to look at the time budget for how much time they spend grooming. So this is the time, this is a percentage of their day. And these are the three experiments. This was the behavior where they're by himself. The social was the one that I mentioned briefly. There were the two crabs in a tank divider. They cannot touch, but they can see each other, have pheromone contact. And then agonistic was where they're fighting. The sample sizes are listed here. And I went ahead and I put the percentages, their time budgets here, just so if you're more of a read and write learner, you can see it easier. And what we can see is that, yeah, when you give these crabs any possibility of primary behaviors happening, grooming goes way down, right? And there was a significant difference with a p-value of less than 0.05, right? And so the time budget did decrease. The crazy thing is, guys, in all of my shrimp studies that I've looked at, and there's been several of them, that shrimp groom themselves about 25% is their time budget. And we can see by themselves, the highest time budget that I have is 5%. So crabs are grooming themselves much less than shrimp are. And to put this in numbers for y'all, is that the individual time budget is 5.22. So what that means is this poor crab grooms itself for 1.25 hours a day, which is 75 minutes. You give that crab the presence of another individual, it goes down to 10 minutes, guys. And why is this important? These crabs, they live together. I can go out and collect at nighttime 100 crabs in an hour. And so these crabs live in really, really high density in their areas. And so because they're living in high densities, their environments are more like this. And so these crabs are probably not grooming very much out in the field, right? Which has very high implications to our water quality. So summary of results, grooming frequency and time was not related to the size of the individual. The 3M was the most used grooming appendage with that palp that goes up and scrapes. The body regions were respiratory and sensory, not decorating. The grooming time budget decreased due to primary actions, and it was much less than what I thought the time budget was going to be less. So y'all ready with your phones? Okay. I hope y'all do better on this one.
Now they need to pay attention.
Yeah, I guess I should have said that in the beginning. So crabs will groom regions of the body where they decorate. And I just reviewed all these, so you should do pretty well. So y'all are right that crabs do not groom regions where they decorate, right? So that hypothesis was clearly rejected. My second one's larger individuals groom their bodies more than smaller ones. And remember, I predicted this because larger crabs will have a terminal molt. And so they won't get that refresh of molting the next time. And again, guys, you're right. This one was rejected. There was no relationship between carapace length, carapace width, and grooming. So 3, the grooming time budget will decrease with the presence of another individual, which would mean that it's secondary, that grooming only occurs when primary behaviors really are not present. You're right on this one. This is actually the first study that has actually documented that grooming is actually a secondary behavior, right? And so that one is accepted. And one thing to remember, guys, is that these crabs are really not cleaning their bodies very much, which means that this crab is, if we're using this as a model for other crabs, these crabs are going to get fouled really fast, okay? So now I'm going to go into morphology, which is short and easy. So the deal is, my problem is that, so what I would predict is that these crabs should have different morphology to allow them to, if they're not grooming themselves for very long, maybe they have special super seedy that if all they do is have to scrape once and it really does an efficient job in grooming. So maybe they have special morphology to not only be efficient in grooming, but also maybe to actually groom those decorating seedy. And so my objective was trying to determine if they did have some sort of special seedy that's not seen in other decapods, right? And so what I predicted is that they would have a lot of different seedy to clean themselves externally on the outside as well as internally, because remember, their gills are on the inside. So my methods is that I went through standard SEM procedure. For every appendage, I looked at 4 different appendages. And what I did is I looked at top and bottom, lateral and medial, proximal and distal. So all around the appendage was looked at in the SEM. We have an SEM at UT, so that's the one that we used. And for most of them, I put them on a stub to look at because they were small. But the big pinchers were so big, they clearly fit on a stub. So we have like this clamp, like a vise that we use. So we had to do that one a little bit differently. And I said the microscope is located at UT. So first of all, some morphology. This is Mr. Crab. This is his face. These are his eyes. These are his mouth parts. And what you see from this crab is fouled. Okay? He is way dirty. And these are his mouth parts, and I'm enlarging it. So this is the deal. This is the third maxilliped. I'm going to work you all through this to make sure. The appendages that we looked at in the morphology study are the appendages that were shown in the grooming study to actually be used for grooming. So the first one is the first maxilliped. It's mostly a feeding appendage to help manipulate food, but it is sometimes used in grooming. Very rarely, but it is. The second maxilliped, which I've outlined, which is a totally different shape, which is used in feeding, but also in grooming. And then we have the third maxilliped, and guys, I'm going to call this the square, okay, because it looks like a square. The deal is that it's a combination of technical terms, the endopod, which is right here, then this little sliver of a piece, the exopod, but I'm just going to call it the square, okay? And then we have this triangle. This is this region that we talked about water going into the gill chamber. And this is called the protopod. I'm going to call it the triangle. Okay? And then I have the palps, which are the little flippy things that flip up and clean the antennae, right? And then the other things that I looked at, these are the epipods. Remember how I said when the maxillipeds go like this, it cleans their gills? This is a picture of the carapace removed on a dead crab. And then the epipod right here. And these are the gills. And this is one of them. And there are 2 other epipods that clean the gills underneath the gills. And then I have the pincher I also looked at as well. So some results. We found a ton of different types of seedy, okay? The first one that we found, we're just going to call Pappas one. What Pappas looks like is it kind of looks like a long, thin pencil with little projections coming off. The little projections that you see here, guys, are called sexuals. Pappas 2 was fouled completely, but it has a lot of sexuals kind of going all the way around it. So you can see this is one seedy here, and there's stuff going all the way around, but it's also fouled a lot. Simple seedy are really boring. There's nothing on it morphologically fun. Simple seedy are usually associated with chemosensory. And then we have serrate seedy, which are my favorite. They look like they have big teeth, okay? So serrate seedy type 1, what it is you have 2 rows of teeth. You have row 1 here, row 2 here, then there's a groove in the middle. So you can see the groove that comes along right in here. Serrate type 2 is right here. You can see it still has the little teeth. The teeth are called denticles. But these little teeth, and there is a groove in the middle on type 2. Type 3 is really rocking. I'd never seen type 3 before. Type 3, the teeth or the denticles were really big, and the teeth interlocked. And so was there a groove? Yes, but you could not. You can see the teeth interlocking, and you can see underneath that there is a groove there. And then serrate type 4, again, I had never seen before. And what it is it looks like simple seedy, but there's a thin row of teeth here and a thin row of teeth on the other side, but there's no groove. And then cuspidate is also really rare seedy. It's different from simple because it's thicker and morphologically shorter. So what does all that mean? So on the palp, remember the palp is a thing that was flipping up going like this. What I did is this is the palp right here. This is the end of the palp, and I'm not going to go over the morphological structures here. But when I enlarged the palp, what I found was serrate seedy type 1. And what we can see here is that, yeah, there's a little bit of fouling going on, but for the most part, it's not horrible. Some of the teeth, the denticles, are broken off, but it's not too bad. when I go over the square, so this would be the square, it's kind of upside down. This is going to be the endopod, the exopods. This is the endopod, exopod. That's where you're looking at. And we can see Pappas type 1 completely fouled. We can see also we have serrate type 1, all the big teeth there. You can see the groove. And we can see simple seedy. So also along the median region, so kind of along this region, what we see is we see two different types of seedy, and then we see lots of fouling. So this is Pappas type 2, y'all. You can see some of the sechels coming off, but for the most part, it's fouled ridiculously. Remember that we saw this consistently on many different individuals, okay? We see Pappas type 1 and then simple seedy coming out. I actually love this picture. Okay, so now if I look at the triangle, what are we going to predict with the triangle? If that triangle is where water is coming in, this triangle better be loaded with seedy. It better stop any sediment from getting in my gill chamber, because the last thing I want is sediment getting stuck in my gill chamber, because it'll mat down on my gills and it will be it'll decrease respiratory efficiency. And what we find is exactly that. Okay, so this is the triangle right here is right here. This kind of goes back a little further. What we can see, enormous amounts of fouling, and then we have these really long type of seedy. Y'all, this is serrate type 1. Y'all can't make it out. One, you can barely see the teeth. The groove is completely chunked full of sediment and debris. So that means it's really fouled, which is probably really good for the crab, because that means it's probably catching the stuff before it goes into the gill chamber. This is Pappas type 2. You can see the long, thin pencils, the sechels coming off. It's matted down. It's fouled. Okay? So now if I go into the epipods, which are the gill structures, okay, they need to clean the gills. Okay? When we look at this, guys, they're beautifully clean. On every individual, they're clean. Okay? Which means that triangle did its job, right? And so this is the end of the epipod, so the end kind of in here. And what we see is we have several different types of serrate seedy. We have type 3. You can see some debris in the groove, but all the teeth, the denticles, they're perfectly fine, right? We're good. We can see serrate type 2, not fouled at all. Here's the groove on serrate 2. Again, not fouled, not clogged up. This is serrate 1, again, not fouled, okay? So now we go into the periopod, which is the pincher. And what we see, no seedy, perfectly clean, which makes sense because these chelipeds are used for feeding, and they're going to pick. These chelipeds, they don't brush, they don't scrape, they pick. So there's really no seedy on it. The only seedy that we found was little tufts of seedy here, which basically what these guys are, these are simple seedy, and when you cut them in half, it looks like this, and there's an opening in the middle that you can see. That means it's for chemosensory. Okay, so that's pretty fun. So some conclusions, guys, is that I know I'm a girl and I am very girly. Okay, the deal is though, is that we can